is advised.
Breaking news, Wildcats. There's a serial killer on the loose, and his name is Mr. Giant Tumor, and he is armed and very dangerous. Please call 911 if you see him or know anything about this suspect. Police fear he may be able to alter his appearance and or create new paths to avoid capture. Okay.
Catch me if you can. I down-regulate my MHC to avoid the T-cell police. I create shortcuts and stuff to avoid them.
Have you guys started studying for pharmaceutics?
Yeah, I just received an UA alert saying that there is a serial killer on campus by name Mr. Giant Tumor, and he has a shared MHC1.
Oh my gosh, I think I just saw him running by. Let's go call 911.
Yeah,
oh my
let's
god.
call 911.
What's your emergency?
Hi, I just saw a guy on campus, and it looked like Mr. Tumor. I think he's on the most wanted list. He seems to be changing his appearance. Like, he was wearing this MHC1 tag, and he seemed to throw it off, and I just wanted to report it. It looks like he was running away.
Okay, we're sending forces over now.
Okay, thank you. About what we're gonna do.
Alright, everyone calm down. Investigation team here. What's the situation?
Okay, so I saw Mr. Tumor, and he was running away towards the bathroom, and I think he went into the lung room.
Okay. Got any information on him?
I got it!
Alright,
I think I know where he is!
We got a tracker on him.
Okay.
We're gonna go back to the office, we're gonna devise a plan, we're gonna kill him, alright? We're gonna
Okay.
kill him dead.
Sounds good.
Okay.
Thank
Let's
you.
go, team.
I forgot to ask you the questions.
Oh, okay.
So what did you see again?
So I saw him running towards the bathroom. It looked like he went into the lung room.
Right, my boss is gonna give him that.
And so he had an MHC1 marker, and he seemed to strip that. And I noticed
MHC?
MHC1.
Okay.
And then it also had IL-6, TGF beta, and TNF alpha on there.
Okay.
I just couldn't recognize all the stuff. There's a lot of stuff on there. I think IL-10, interferon gamma. That's just all I have for you. I'm sorry.
Okay, thanks.
Thank
Back to my
you.
boss.
Hi, IT. So what did you guys get from the investigation concerning the serial killer?
We're gonna blow up the whole building.
Whoa, 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 whoa. Stop the bus, okay? We know where he's located. The nano tracker says, it says right here. He's in the lung room.
But we've always done it this way.
This is the best way, why overcomplicate it? This is the best
Hold
way.
on. Let's put our ideas together, okay? We know this guy is dangerous. He's a serial killer. We have to be careful, and we know that there might be civilians in that room. So we have to come up with a strategy.
Exactly.
Not to blow up the whole building.
The strategy, there's this new way of killing Mr. Tumor. I swear. It's this new theory, and it's called Trojan horse therapy.
Alright, everyone, this is Team HSG. This is Agent Virus, and this is Agent HSG. HSG is heparin, sulfate, glycans.
That's me.
Alright, so the plan is we're going to load this T cell with some virus. The virus is going to be protected by HSG.
With a coat.
They're going to go in, our tumor's going to have no idea what's going on, and it's going to see these guys as friendly. Tumor's going to release these two, and then the virus is going to kill that tumor cell. Alright, everyone got it?
get down to some business.
Hey, I found my T cell.
Okay, I'm gonna come coat you now.
Hey, let's go take down the tumor.
Woo!
Men's bathroom.
You comfy in there, tumor?
Why? Yes, I am very comfy in here. How are you guys doing?
Ready
Well.
to take you down.
I've been waiting for you.
We've been waiting for you. We look forward to destroying you.
Gang
Gang
cycle
Cycler, we're ready to destroy you. Gang Cycler, come over here.
Where's Mr. Timo?
Oh, you're too strong for me. Give me a cycle here.
I got Mr. Timo.
Make me a cycle here. You're too strong. Oh.
Alright guys, today we're going to go over some real basic stuff, so let's get to it. We're going to talk about some T cell circulation as well as tumor cell death. So basically, T cells are circulating throughout the body. They patrol the body, they're just moving through the blood and the lymph, etc. They can even penetrate tissues in the blood-brain barrier. Check out that brain, it's pretty sweet. And then next, it'll mount a response against a tumor. So we're going to go through the cell death real quick. So there's a tumor. It's pretty upset. He's expressing the antigen, and the circulating T cells will recognize that antigen, and they'll go through their signaling pathways like we've learned many times. The MHC class 1, the TCR, the B7, the CD28. The T cells will produce cytokines such as TNF alpha, TGF beta, IL-2, and more. That will recruit and activate additional T cells, which will then lead to killing that ugly tumor. And so that's the basics of the circulation and the cell death.
Let's go.
Hi guys, so I'm going to explain kind of why we use the Trojan horse mechanism and we combine T cells and retroviral vectors. So to start, so the Trojan horse combines these two mechanisms because they can't work by themselves. So T cells, they can only be used for the treatment of an infectious disease, not tumors, and they're unsuccessful against like specific tumor antigens that we need to target. And then retrovirals are restricted by their tissue type. They can't act against like many different types of tissues and the diversity that we see within that tumor cells can attack and be. And then also they rapidly get neutralized within like the systemic circulation, so that isn't good. And then they can't get into the tissue that we need to target, and toxicity of the patient can also occur. But when you combine these two mechanisms together, you get the Trojan horse therapy, which is a successful therapy that can be used against tumor cells.
So what is Trojan horse therapy? It's going to combine that tumor target T cell and the gene therapy. So here's my tumor-specific T cell with my retrovirus. That is coded with HSG. Anyone want to guess what the H stands for? Phoenix? Tucson? Any thoughts? Heparin. I don't know if I'm spelling this right. The S, anyone? Sulfates. Glycosaminoglycans. So, when the T cell is seen by the tumor cell antigen and it's recognized, the retrovirus vectors are released into the tumor cell. Those vectors encode two proteins, IL-12 and herpes simplex virus thymidine kinase. IL-2 is going to provide proliferation of the T cell with the specific receptor. The herpes simplex virus modifies the prodrug ganciclovir to a toxic derivative, destroying the tumor cell. Oh, and this is Brittany.
All right, so in the first model, we have these T cells, all right? And they came from this organism over here, right? So we take the T cells, but the DNA that's in here actually came from somewhere else, a different organism. These T cells are specific to a protein that we call ovalbumin, okay? Ovalbumin, okay? And these lung, this is the lung tumor. And the lung tumor expresses this protein, ovalbumin. When this connection is made, T cells accumulate in this lung tumor. And eventually, the tumor shrinks. So now it's small. Yay. But in lung tumors that don't have this protein, ovalbumin, there's no T cell accumulation. So it stays the same size and it can actually grow. When this lung tumor, with our T cell accumulation and the expression of ovalbumin, expresses IL-12, IL-12 enhances T cell proliferation. So you get even more shrinking of the T cell tumor, of the tumor, not of the T cells, but of the tumor.
In the second model, when the tumor dose was increased, we had neither the tumor-specific T cells or the cell-free virus. None of them could produce complete remission. So there was no complete remission with this one. But if the T cell was loaded with tyrosine kinase expressing retrovirus, these T cells were able to cure 6 of the 9 mice infected with the tumor, and the 6 mice were resistant to tumor re-challenge.
Hi guys, so we can, there's a more effective way of killing tumor now. We can combine the antibody IgA with IgG and come up with a cross-type antibody which is called IgGA, and that's more effective at killing tumors. So I'll go ahead and draw that out for you guys so it makes more sense. So we'll use different colors. This is IgA. If I could get my pen to work. Okay, we'll just use this. And that'll make which is more effective. So yeah, so this is more effective than killing the tumor than these two alone.
So there are different approaches to the Trojan horse. We've already covered a few, but I wanted to cover 3 more. approaches. And so the first one is actually, we're going to go ahead and destroy the tumor. So the tumor is destroyed by our local therapy against the cancer. And the resulting tumor particles are then utilized as a vaccine against a tumor. So that's one way that we overcome the tumor. Another way is we can alter the cytokines and thus change the environment of the tumor to allow for it to be killed, to kill tumor. And so what we would be doing there is we downregulate IL-6 and TGF beta and IL-10 and VEGF, while also upregulating IL-2, IL-12, and interferon gamma. So then the last approach is just overcoming the dendritic tumor cells. So overthrow dendritic tumor cells, which allows CD8 to enter tumor. And so it turns off regulatory and suppressive cells. So reg cells. So that an example of that would be the anti CTLA-4 and the Tregs. So those are just some approaches as to how we could overcome cancers of the body. And then should I just put Claire? Or no?
One. We have a unique method to target tumor cells, but what does this mean for the future? What exactly is the goal? The goal is to have a more widespread application for this therapy. And how can this be achieved? Although, as for now, it specifically targets tumor cells, what we could do is have multiple retroviruses per application. Something else we can also have is use more transgenes. And finally, the final goal of is to be able to use this in non-malignant settings, basically broadening its therapeutic uses.